Hello, my name is Mark Walker, and I'm a marine biology graduate student at Texas A&M Galveston. In this presentation, I will be discussing my research paper on plastic contamination's effects on marine wildlife and ecosystems. There are many dangers of plastic pollution, and it is an enormous threat to the world's oceans as it greatly harms marine organisms and ecosystems. More than half of all marine debris is plastic, and it is estimated that if the current rate of plastic pollution in the ocean does not change, there will be more plastic than fish by 2050. One of the biggest challenges with plastic is that it is not biodegradable and it is persistent in the environment. Additionally, sunlight will break it apart into microplastics, which are defined as plastics less than 5 millimeters long, and these are difficult to remove from marine ecosystems. They are also often confused for food by marine animals, especially fish. Consumption of microplastics can lead to an avoidance of real food and consequently starvation. Internal blockage is another issue from consumption of microplastics. Plastic pollution in the ocean is also responsible for the deaths of countless marine organisms through both entanglement and poisoning. Plastic debris can also transport small invasive species, including amphipods and isopods, leading to disruptions in many ecosystems. Plastic debris also contains many harmful chemicals, such as persistent organic pollutants, better known as POPs, heavy metals, and pesticides. These chemicals can have a variety of potential harmful effects, including death. POPs have been known to cause cancer, endocrine disruption, and adverse effects on reproduction. Heavy metals such as mercury can cause negative effects on development and reproduction, and they can also inhibit enzymes involved in important processes such as cellular respiration. Pesticides like DDT can cause metabolic disruption, reproductive issues, and reduced growth. These chemicals are difficult to remove from marine ecosystems and can also bioaccumulate in the tissues of marine organisms. Not only does the buildup of plastic contamination in the ocean harm individual marine organisms, plastic pollution also has many adverse effects on marine ecosystems as a consequence. Through biomagnification, toxic chemicals can increase in concentration in higher trophic levels of a food chain. Lower trophic level organisms such as fish often consume plastic and absorb chemicals in their body fat and organs as a result. When fish are consumed by predators like seals, birds, and humans, biomagnification occurs, resulting in a high concentration of toxic chemicals in the tissues of higher trophic levels. Many keystone species are higher trophic level animals, and they can end up with the highest chemical concentrations in their tissues. Animals at higher trophic levels typically are smaller in number and reproduce slowly, so biomagnification is a huge threat to many of these organisms. Many important keystone species, like dolphins and sea otters, are essential to maintaining balance in their ecosystems. Furthermore, contamination of marine ecosystems concerns human populations as well. A sixth of the animal protein that the human population consumes comes from the ocean, and biomagnification can occur in humans as well. So the objective of my research paper was to investigate how plastic pollution is affecting marine life at the individual level and the ecosystem level. My thesis was that due to widespread plastic pollution in the world's oceans, marine ecosystems are threatened, which could lead to disastrous outcomes for humans and wildlife alike. My first case study was on Akupa weakfish, and it investigated plastic contamination in the fish species, which is an economically important species in a Brazilian tropical estuary. The results found that the weakfish often ingested plastic debris as well as accumulating plastics from contaminated prey. 530 weakfish specimens were sampled, with most of them being juveniles, and the researchers found that 64% of the fish at all ontogenetic phases had plastic debris in their stomachs. 100% of the adults sampled had plastic debris in their stomachs as well. Plastic debris was more commonly consumed than any other food source, as amphipods were the second most commonly digested item at just 33%. In addition, 97% of the plastic contaminants found in the weak fish digestive system were microplastics. Plastic debris was found in greater than 50% of the fish sampled, regardless of the season or area that the fish were sampled in. Due to the economic importance of this fish, the amount of microplastic contamination in their tissues is a great concern. The second case study also focused on fish and investigated plastic contamination in myctophid fish, also known as lanternfish, in remote areas of the South Atlantic Ocean. The area studied was a previously unsampled gyre where there was a large amount of plastic debris. 3 different zones within the gyre were sampled, and the amount of plastic debris was found to be similar in each area. Many different chemical contaminants from plastic sources were found in the fish sampled. Both polychlorinated biphenyls, or PCBs, and polybrominated diphenyl ethers, known as PBDEs, were found in several water samples that were taken. The results found that myctophid at areas with higher plastic density had a greater concentration of PBDEs in their tissues. PBDEs are commonly added to plastics as a flame retardant. Other chemicals measured, such as PCBs and bisphenol A, were not correlated with plastic density at the sampling area. However, this study indicates that increased amounts of plastic in the area will also correlate with increased amounts of plastic contamination in fish tissue. With the increasing rate of plastic contamination, this poses a great risk to marine wildlife everywhere. The third case study I researched investigated plastic debris ingestion in Franciscana dolphins off the northern coast of Argentina. 106 dolphins were sampled, and 28% of the dolphins had plastic debris in their stomachs. 34% of the dolphins in estuarine environments had ingested plastic debris, while 19% of dolphins in marine environments had ingested plastic debris. The type of debris was similar in both environments and consisted mainly of packaging material such as cellophane, bags, and bands. Fishing gear and line were also one of the main sources of plastic debris in the environment. Dolphins that were recently weaned had the most plastic debris ingestion. This is likely because an increase in entanglement in gill nets and also bioaccumulation of heavy metals are most commonly found at this ontogenetic stage. For conservation purposes, young Franciscan dolphins in the first few months of independence from their mothers should be considered the most at risk. The fourth case study focused on diving seabirds in the Arctic. And then although the Arctic is in a remote location and far from major sources of plastic production, plastic debris ingestion is found in many marine organisms in the Arctic. In this case study, researchers investigated microplastic contamination of both zooplankton and their predators, little auks, which is a type of Arctic seabird. The study was first conducted off the coast of East Greenland in 2005 and conducted almost a decade later, again in 2014. Due to the increased rate of sea ice melting, the environmental conditions in 2009 and 2014 contrasted greatly. Sea ice can serve as a sink for microplastic particles, so the melting of sea ice can release a large amount of plastic debris into the ocean. There was also a large increase in plastic production worldwide between 2009 and 2014. The results of the study found that the concentration of microplastics in zooplankton sampled in 2014 was 2.4 times greater than the concentration in zooplankton sampled in 2009. Interestingly, the concentration of plastic debris in seabird diets actually decreased slightly from 2009 to 2014, although not significantly. 44 birds were sampled in both years, 2009 and 2014, and 100% of them had consumed plastic filaments. Additionally, little auks had a significantly higher concentration of light-colored plastics in their diet than dark-colored plastics. This suggests the birds frequently mistake light-colored microplastics for their natural prey. The tissues of seabirds bioaccumulate through both direct consumption of microplastics and through biomagnification as they consume contaminated prey. As the rate of sea ice melting continues to rise, large volumes of trapped plastic debris is expected to be released in the Arctic. This will cause Arctic marine organisms to be increasingly more vulnerable. In this case study, researchers investigated the amount of anthropogenic debris in fish and shellfish at food markets. Seafood was sampled from markets in California and in Indonesia. In Indonesia, plastic debris was found in 55% of all species and 28% of individual fish. In California, 60% of all species sampled were contaminated, and 25% of individual fish contained anthropogenic debris. All of the debris that was recovered from seafood in Indonesia were composed of plastic, while the debris composed in California were mostly made of textile fibers. The differences in type of debris is likely because of the varying sources of production and methods of waste management between the two areas. The United States has a more advanced waste management system and misplaces significantly less tons of plastic than Indonesia. This is likely why plastic debris was much more prevalent in seafood in Indonesian markets. The presence of anthropogenic debris in seafood available for consumption raises the risk of biomagnification of chemicals through seafood ingestion. The researchers conclude that more studies are needed to assess toxic effects of anthropogenic debris contained in seafood. This case study focused on the after effects of the 2011 earthquake in Japan. In 2011, when the enormous earthquake struck in East Japan, it generated a tsunami that transported many organisms on plastic objects across the Pacific Ocean. Researchers investigated this phenomenon and found that 289 Japanese marine organisms had been dispersed from the coastal area. The vast majority of the marine organisms documented were invertebrates such as mollusks and crustaceans. Some species of fish and protists were also discovered to have been spread by the tsunami. Since plastic is non-biodegradable, marine organisms could be attached to them for a long time and were transported as far as the shores of North America. This was the largest documented dispersal of coastal species by rafting events. Since climate change has caused the rate of severe storms to increase, the amount of mass plastic debris dispersal events are expected to increase in the future. The transport of plastic debris has also caused invasive species to spread to new areas and disrupt ecosystems. The transport of plastic debris through the world's ocean systems presents a major threat to ecosystems worldwide, and the problem is expected to become more critical as climate change and plastic production rates continue to rise. So to synthesize all of the information from the case studies, a reoccurring theme throughout the studies was that the amount of plastic debris in marine ecosystems continues to increase, and marine organisms worldwide have plastic debris ingestion regardless of the area. Even in the remote Arctic, 100% of the seabirds sampled had consumed plastic debris. Furthermore, the amount of plastic debris in Arctic waters significantly increased between 2009 and 2014. The amount of plastic debris in marine environments will continue to grow as sea ice melts and the rate of plastic production rises. Massive storms are expected to become more frequent and cause a greater dispersal of plastic debris. In addition, Plastic trash will continue to accumulate in massive ocean garbage patches, especially in gyres, and organisms will be in danger of getting trapped in this garbage. Marine animals can become contaminated by plastic through both consumption and entanglement. They're severely at risk of being entangled in plastic nets and other debris. The increasing rate of plastic pollution in oceans will make it more difficult for marine ecosystems to avoid contamination, and plastic ingestion will likely be found in a greater percentage of animals over time. The increased number of chemicals associated with plastic will also likely result in more health risks, such as the aforementioned development and reproductive issues that occur from POPs and heavy metals. Many marine animals at lower trophic levels, such as fish and birds, cannot differentiate between microplastics and their natural prey. The bioaccumulation of plastic contaminants at lower trophic levels can then lead to biomagnification in predators at higher trophic levels. Once one species in a food web becomes contaminated, this plastic contamination can quickly spread throughout the ecosystem. All ecosystems rely on balance between the trophic levels, and any decrease in one specific marine organism population due to plastic consumption will have drastic effects on the entire ecosystem. Contamination of keystone species such as dolphins would likely have the greatest effect on a marine ecosystem and should be considered the most important conservation. Dolphins help keep fish populations in check and also help round up fish in groups, creating feeding opportunities for many other organisms such as birds, sharks, and whales. A decline in dolphin populations would likely lead to a rise in fish population size and less feeding opportunities for other predators that rely on dolphins. Furthermore, marine organisms are not the only ones at risk, as humans face potential adverse health effects from ocean pollution. Seafood is a large part of humans' diets, and the consumption of contaminated seafood can lead to biomagnification in human populations. Humans need to recognize that they're an integral part of ecosystems and that they are affected by plastic pollution as well. The chemicals associated with plastic pollution can build up in humans, and the resulting consequences of these health effects could be disastrous. In conclusion, more conservation efforts are needed as marine life faces greater risk from increased plastic pollution. Due to the importance of keystone species, conservationists should consider concentrating most protection efforts on animals such as dolphins. And plastic pollution does not just affect marine animals. Humans are an integral part of the food web and ecosystems and face adverse health risks as well. These are the reference cited, all the different case studies I used in my research paper. And thank you for watching my presentation. I hope you enjoyed it and learned something new. Thank you.